We're getting into class 2 of Embryology for Colloquium 1. Let me just write it down so that it makes more sense. Colloquium 1. Here we're going to talk about the placenta and we're going to talk about the umbilical cord. Umbilical cord. Those are the only two slides for class 2. However, the placenta is quite a difficult slide and most of this video is going to be dedicated for specifically the placenta, while the umbilical cord is one of the more easier slides. So it's an even balance for this class. Number one thing I'd like to say is in the colloquium, the most difficult thing most students will have an issue with is the fact of identifying the slide that you're at. Not the structure names, but the actual slide. So for the placenta, the structure that we're looking here, I will try to help you visualize it in the best way I can. Like, this is how I made sure to remember that we're looking at a placenta rather than other structures that we covered in previous classes, right? So the placenta has a distinct structure. It has this area at the top known as the fetal side, and this area at the bottom known as the maternal side. And the things in the middle are called the intervillar space, which is all of the white things, and then also the villi, which are all of the pink things. How I was able to visualize this or to compartmentalize it was by imagining it to be like a shore between two separate countries, right? This would be the fetal country or the fetal mainland, and this would be the maternal country or the maternal mainland. Now, in between, we have the sea, in quotation marks, with all of these villi or tertiary villi, all of these small pink stuff, all of those being the villi, but I call them, or I imagine them as small islands. This helped me to identify that we're looking at a placenta in every image. So if we look at these images taken on the microscope, because the placenta is such a big organ, we're not going to be able to have such an easy cross-section seeing both the fetal part of the placenta and the maternal part of the placenta. We will only mainly, like at maximum, have a section like so, looking like this or looking like this, or in the middle maybe like so. This is the maximum we're going to be able to see. So if we zoom in something like this or something like this, etc., right? And this is how it will look like in the microscope. As we can see here, these are not the mainlands, these are smaller islands, also known as but these are tertiary villi. Please don't write islands. This is just me trying to bastardize these structures as best as I can to make them more memorable. But where you're going to write in the colloquium is obviously the tertiary villi. So all of these smaller structures that I call island are tertiary villi. While the mainland we can see on parts like this. Here we can see a big chunk of a mainland, and this being smaller islands, right? So that's how it's going to look like in the microscope. Here we can also see all of these tertiary villi, or islands as I like to call them, and also here them looking like smaller islands, right? So that's how it looks like in the microscope. Going back, again, we're not going to have this big of a magnification. We're going to have quite a small section of it. And for the colloquium, you will have to know the difference between the fetal side of the placenta as well as the maternal side of placenta. And I've written the differences right here. So the fetal part and the maternal part. We have two clear visual distinctions between the fetal part and the maternal part. Both have this pink area that I call the mainland, followed up by these islands, right? Here we have islands, and then here we have the maternal part, which is all of this pink area. Now the two visual distinctions we'll start with. These are what you're going to look after when you're doing the colloquium. in order to be able to identify what side you're on, if this is the fetal side or the maternal side, right? If we look at something like this, are we up here or are we down here in the maternal side, right? In order to be able to see that, we'll need to look at these two important signifiers. The first signifier is the border of either the fetal side or the maternal side. So for the fetal side, we have a clear, simple columnar amniotic epithelium border. Now this is a structure name that you have to remember because this might come in the colloquium, but this is something that exists in the fetal side. Now what are we looking at exactly? We're looking at all of this. Now I'm going to be covering it with my red pen, but you will see it once it disappears. So all of that is known as the simple columnar amniotic epithelium. And the simple columnar amniotic epithelium is very distinct. It's very dark. However, zooming in, if we zoom into this section, for example, we'll have something looking like this. And then the simple amniotic epithelium, columnar epithelium, will look something like this instead. So you can very much see here one of these cells that, makes it very distinguishable as a columnar epithelium or epithelial cell, and you can see how well it is distinguished, how well distinct it is in comparison with the remaining pink area of the fetal part, right? You can see it here when zoomed out, this area or border compared to the remaining pink part. If we compare it instead with the maternal placenta, and we look at its border here, you can see that there's nothing that is really distinct here. No cells that really line it as deeply as we see here, correct? So here we will see absolutely no border, and here we will see a border. And that is the main difference between the fetal placenta and the maternal placenta. Now looking at the demo lab slides, let's keep that until later when we go through the second distinction first. So the second distinction is the sort of content of these parts. So we have the fetal area or the fetal placenta here, and then we have the maternal placenta here. Now the contents are of course cells, right? These cells that are scattered all across here are different from these cells. And I'll tell you why in a second. First of all, the fetal cells in the fetal area is made up of mesenchyme, specifically extra embryonic mesenchyme, which is a structure that you'll have to remember for this colloquium. So the extra embryonic mesenchyme, which is this area right here, is made up of mesenchymal cells, right? You can see them down here as well. And the mesenchymal cells, although they look very oddly shaped at times, they are pretty much evenly formed. There's not really that much of a difference between this cell and this cell, or this cell and this cell, etc. The cytoplasm is about the same. They look very even, they look very uniform. And the texture looks very smooth and not as rough as the texture of the maternal placenta, which looks something like this. See how these cells are differently sized? Compare this cell with this cell. See how huge this one is in comparison to this. This is because, again, the maternal area has a different type of cell compared to the fetal area, or if we're gonna zoom in, fetal area here, that only has mesenchymal cells. Now the cells, again, fetal placenta has the mesenchymal cells, which makes it look a lot smoother when zoomed out, while the maternal placenta has these HACPT cells. Now you must spell these out during the colloquium. What does HACPT stand for? Hormonal active cell of proliferating trophoblast. Let me find them. That's full term. Here we go. Hormonal active cell of peripheral, not proliferating, sorry, peripheral trophoblasts. And they look something like this. They are larger at points, smaller at points. Again, very blotchy, I would say, very uneven compared to the mesenchyme of the fetal placenta. The maternal placenta is very much rough. It looks like if you touch it, it will look a lot bumpier than this, right? It looks like it's covered with acne, sort of. So that's the main visual difference, other than the border that exists in the fetal placenta but doesn't exist in the maternal placenta. It is the texture of the actual pink stuff. The maternal placenta having more blotchy, rough structure made up of the HACPT cells, while the fetal placenta has a lot smoother surface, which is because it's made up of the mesenchymal cells. Yeah, so now that we know the two different visual signifiers between the fetal and the maternal placenta, we can actually start guessing where we are in real microscopy slides. So these are real microscopy slides taken from the demo lab. If we look at this area, this is either the fetal or the maternal part of the placenta. So the fetal part, like I said, has two things. It has a clear, simple columnar amniotic epithelium border, and it's made up of these mesenchymal cells that makes up like an even and smooth surface. So looking at this, do we see a clear, simple amniotic epithelium border here? And how does the surface of this area, this and this are the same slides. This is just more zoomed in. How does the surface of this area look like? Is it smooth? Is it rough? Is it blotchy? Does it look like acne? I would say if you guessed this is the maternal part, you would be correct. Again, looks like acne, right? That's because all of these cells are HACPT cells, and we also have no simple columnar amniotic epithelium. We have no such border that runs across this mainland, right? That's why we know how we know that we're in the maternal part of the placenta. Okay, compare it with something like this right here. Now this is another mainland area, right? These are tertiary islands, as I like to call them. Again, the quality of this image is not that great, but there's one specific signifier that I'd like you guys to guess that will be able to that you'll be able to use in order to determine if we're in the fetal side of the placenta or the maternal side of the placenta. If you guessed the border being super clear and distinct, then you are correct. See how dark this border is? This is what we call the simple columnar amniotic epithelium. And if we see it, we can immediately assume that we're in the fetal side without even having to look at anything else. Again, the texture of the pink part looks smooth, but it might also be because of the quality of the image. But since we can see the border so clearly, we can just immediately assume that this is the fetal side of the placenta. This is a much more difficult slide that was brought up in the demo lab. I doubt that this will be used in the colloquium because it's such a unforgiving slide. If I saw this first glance, I would have said maternal part of the placenta, but it's in fact the fetal part. Why? Well, first of all, even though it looks like the surface is blotchy, it actually isn't. It's just because of, I guess, this blood vessel, it looks a lot more uneven than it should. This is actually the mesenchyme with the mesenchymal cells, and we do see a border here, just very, very, very not distinct. So again, I don't think that they will use this in the colloquium, but just to make sure, this is how the fetal side might also look like. Important part though is for you to remember these two visual signifiers. Now we've talked about the visual signifiers, we will talk about the theoretical differences that you must know for the structure part of this slide. So for example, the pink area for the fetal placenta is called something different compared to the the pink part of the maternal placenta. Now, the pink part of the fetal placenta has a colloquial name known as the chorionic plate. And the chorionic plate is split into two parts. First, the border, which we all know and love, the signifier, known as the simple columnar amniotic epithelium, of course. And then all of this pink area underneath, that I did call mesenchyme, is known as the extraembryonic mesenchyme. You have to write extraembryonic before mesenchyme, or else you will not get the full points. And these are made up of the mesenchymal cells, which look even and uniform, right? While in the maternal part, this pink area, even though they look the same, is called the decidua basalis if we're in the maternal part. So again, if they ask you about this layer, then you first have to know if we're in the fetal placenta or the maternal placenta, and you have to and in order to do that, you have to first compare if we see a border or not, and the texture of the pink area. As soon as you figure that out, you can actually know that it's decidua basalis and not chorionic plate, right? And the decidua basalis, thankfully, it doesn't have any layers like the chorionic plate. So the chorionic plate has these two. The decidua basalis is its own thing, right? And it's made up of the HACPT cell, and you have to actually spell it out. And they look uneven and blotchy, which is another signifier, right? The final thing about the difference between fetal and maternal placenta is something that is a little bit more complicated. It's in fact the fibrinoid. And the fibrinoid, if we look at this part, it's a little bit more visualized, I guess. So look at something that is very deep pink or deep magenta color. So all of this area, this and this, all of that, all of these, this, that's all known as the fibrinoid. Now, fibrinoid has different names or different, I guess, prefixes, depending on where the fibrinoid is. Now, there are three types of fibrinoids. There's one that is directly underneath the fetal placenta, which you may see here, like all of this that is lining directly underneath the fetal placenta is known as the Langhans fibrinoid. If we want to see it zoomed in, it looks something like this. So this is the fetal part of the placenta with the chorionic plate. And we have the extraembryonic mesenchyme here. We have that border known as the simple columnar amniotic epithelium. And underneath the dark pink area, all of that is known as the Langhans fibrinoid. You will also see it here, right? While underneath the maternal area, now it's not as visible here, but I'll just imagine that you see a lot more pink than you actually do, or dark pink. This and this would be known as the Nitabuchs fibrinoid. We can see another example of it if we look at the maternal part here, maybe. So see all of this darker pink that is lining underneath the maternal side of the placenta, that is known as the Nitabuchs fibrinoid. So underneath the fetal placenta, known as the Langhans fibrinoid, the maternal placenta, known as the Nitabuchs fibrinoid. This is why it's very important for you to know if we're looking at the maternal or fetal part of the placenta. Okay, and then the third type of fibrinoid, if you see all of these tertiary villi, all of these islands, right, that are tertiary villi, don't write islands in the colloquium, again, I beg of you, also have these pink fibrinoid surrounding it. So all of that isn't directly connected to the line that is underneath the chorionic plate. Here we can also see it kind of here and here. That is known as the Rohrs fibrinoid. We just love men's surnames being used to name the structures, right? So again, Langhans being directly underneath the fetal placenta, Nitabuchs being directly underneath the maternal placenta, and Rors surrounding all of these tertiary villi. Another important thing about the fibrinoid is that a lot of students make the mistake of they assume that this is also fibrinoid, and I would like you to see the difference in color between this and this, right? See how much red this area is, and see how much pink this one is? Well, that's because here we're actually looking at blood, while here we're looking at fibrinoid. Fibrinoid is tissue, blood is of course made up of red blood cells. So don't make that mistake, because that may appear in the colloquium where they ask you about the blood instead of the fibrinoid. And even then, there's a difference between types of blood that we see in this structure, and we'll get to that in a second. But yes, don't make the difference or don't make the mistake of accidentally naming this fibrinoid when you're looking at blood, especially when you can see actual fibrinoid and the color of the fibrinoid compared to the blood color. That is a lot red, right? This is also blood. Okay, that was the difference between fetal and maternal placenta. Now we have another distinguishing area of the slide, which is the difference between new placenta and old placenta. So new placenta and old placenta, they cannot exist in the same slide. It's you're either looking at new placenta or you're looking at old placenta, and for that we don't really take into account the main lens, right? The main lens is not going to tell you anything about if you're looking at new placenta or if you're looking at old placenta. We're actually looking at the area in between, the C area, right? Made up of all of these tertiary villi, okay? New placenta and old placenta are distinctly different in one main area, and that area is what borders these tertiary villi, all of these islands, what cells border them. So we can see them here, and we can see them here. This is new and this is old. Now, for the borders of the tertiary villi in new placenta, we can see that they have two distinct cell layers. The first one being a lot lighter, composed of the cytotrophoblasts, and the second one, the outer layer, being a lot more darker, composed of the syncytiotrophoblasts. And they're very distinct cell layers. You can definitely see them, having a border. If we compare it with the demo labs, here, we can see here more zoomed in, but this is zoomed out, and in the colloquium they will zoom in for you, so you will see the borders. So even if you write if it's the age of it, old or new, at first, and you change your mind once zoomed in, you can just change your answer. But here you can see this inner layer being a lot lighter in the new placenta, this being the cytotrophoblasts, and the outer layer being a lot more darker, syncytiotrophoblasts. Also, these two names you have to know. But again, these are also signifiers of the new placenta as well. Compare it with old placenta. Now these are two images of old placenta. These are two images of new placenta. Do you see a difference here? The pain about the practical part of the colloquium, the worst part of it, is that you will not have a clear, you will not be given both the new placenta and the old placenta at the same time. You will be given either one or none. So if you have this, you're gonna have to know the difference between new and old in your mind. You're not going to be able to compare it with this, because if you had both of these, you would much easier make a conclusion that this would be new and this would be old. But since you're only going to get one of them, it's going to be a little bit more difficult than that. So in old placenta, instead of having these two distinct cell layers bordering the tertiary villi, we can see here that we barely have any cell layers bordering. Maybe here, sometimes we don't even have any cell layers bordering. we can kind of see it here, not really. These are called the cytotrophoblast remnant. The cytotrophoblast remnants. And this is because the outer layer, the syncytotrophoblasts, is degenerating, so it's completely disappeared. And the cytotrophoblasts are in the midst of degenerating, so they are sometimes still there, you can see them here, and then sometimes they're barely even on the border of the tertiary villi. Like you can see here, there's no border here. It looks like it has been completely cut with a scissors, right? Those are the two differences between the new placenta and the old placenta. You can see it more clearly here. The borders here having two very, very obvious cell layers, and here barely having one, if any, right? Okay. Another difference that I would like you to take with a grain of salt, because again, it's hard to know when you don't have both of, when you don't have an old and a new to compare. It's very hard to go according to this next signifier, but I'll mention them anyways. Hopefully it'll help. But again, the most important one are the cell layer borders, and this is the least important one. Might give you a heads up, might not, might also be your downfall. But it is that in new placenta, it's generally lighter, there's more intervillar space, and there's less ros fibrinoid. What is ros fibrinoid? Like we said, ros fibrinoid is the fibrinoid that surrounds the tertiary villi, not the Langhans and not the Nitabuchs, but the one that surrounds the tertiary villi, right? And we can definitely see more of the ros fibrinoid here compared to here, where we can't really see any ros fibrinoid. No, this is blood. This is also blood, I think. This is blood as well. Why do I know that for certain? It's because these are very, very, typical blood cell shape. So you can definitely see that this is blood and not fibrinoid. But here we have both blood, which is a lot more red, and the ros fibrinoid. Yes, so again, it's hard to say if it's, because it's lighter or because it's darker. If we compare it here, for example, if you only see this in on your microscope, you assume that, we can see a lot more intervillar space, which is all of this white area, but that doesn't necessarily mean that it's new placenta. In fact, this is old placenta. So again, I would say the most important signifier are the cell layers. So here we can definitely see a thick cell layer or cell border here as well with the two cell layers, and here we can't really see that at all. We can barely see the remnants of cytotrophoblasts. But I think that's it for the difference between new and old placenta. Now I would like to mention some other things. First off, some other structures. Well, we can also talk about how again we can find a little bit more about old and new placenta, or we can find some examples of it. This would be an example of old placenta. Again, it has more ros fibrinoid here, and also this border of the tertiary villi doesn't look like it's not thick at all, and also it doesn't look like it has more than one cell layer. It looks like it barely has one. So these are the cytotrophoblasts remnant. I think that's it. That would be old. This I think would be old as well, but it's hard to see without zooming in. Even though there's a lot of intervillar space, white area, we can barely see any cell border layers here, so definitely would say that this is old. This would also, I would say, be old because of the cell borders. This too.
See how the cell borders barely has any cell layers? This as well, only one-ish. Sometimes it doesn't really have the borders. The new placenta looks a lot more a lot more different than the old placenta. You can see it here. This is definitely new placenta. You can see a clear cell border, and if we zoomed in, it would look something like this. So once zoomed out, it might look like this, and then zooming in, they will show you that there are two clear cell borders, and that way you will know that it's new, right? Here's another new placenta. Well, it's a dead giveaway, but yes, 2 cell layers here. And of course, getting to old placenta, where we only have 1 cell border, barely that. Yeah, and that's the umbilical cord. Okay, perfect. Now getting into other structures. One of the structures I wanted to mention, other than the HICPT cells that is in the maternal placenta that I've talked about, all of these cells, they can also bunch up together and conglomerate, and in that case we have to write conglomerate of the hormonal active cell of peripheral trophoblasts. So sometimes they are bunched together like so, and therefore you have to write it like so. It's very much reminiscent of what you wrote during the human sperm as well, what we talked about. We could have a conglomeration of human sperm or spermatozoa there. So these are I think the only 2 times you will use the word conglomeration or conglomerate in regards to cell types for this colloquium, I think. Proliferating bud is one of the easier structures, thankfully, in this slide. And they are just very, very dark areas within the slide. So all of these dark, almost black areas are proliferation buds. You can see them here as well. That's a dark splotch, that's a dark splotch, that as well. All of those are proliferation buds, and they are visible everywhere. You can see them here, this one as well. So that's a kind of easy structure. Oh yes, now we're getting into the blood. So we have 2 types of blood in the placenta. We have maternal blood and we have fetal blood. Maternal blood always resides within the intervillar space. See all of this green area? So not inside of the villi, but outside of it. You can see how the blood diffuses within the intervillar space. And what do I mean by intervillar space? I mean all of this white area surrounding the tertiary villi, the intervillar space. That's where the maternal blood resides, while the fetal blood comes from the umbilical cord and runs all the way down into the tertiary villi. The fetal blood and the maternal blood cannot mix. It would be a huge immunological issue if they would actually mix, but in this case they are kept separate. The fetal blood is within the tertiary villi, and the maternal blood is within the intervillar space. Now we can see some examples. I would say a good example where I can show you specifically. Yeah, I guess so. So all of these brown areas are blood. And this brown area is within the intervillar space, within the white area surrounding all of these tertiary villi, right? And if we're looking at blood within the intervillar space, that would be the maternal blood. While blood within the tertiary villi that have these blood vessels inside, once they contain blood, that would be the fetal blood. So the fetal blood may never mix together with the maternal blood. That's why the fetal blood is kept within the tertiary villi, while the maternal blood is kept within the intervillar space. I believe there's a better image up here. Yep. So you can see, as I said before, these are all red blood cells, very characteristic red blood cell form and shape. And these all are within the intervillar space, within the white area. So if it's within the intervillar space, then this would be maternal blood. And fetal blood would be within the tertiary villi. So a good example of that would be this tertiary villi. You can see that there's a blood vessel right here, And then zooming in, we have some red blood cells. Now that would be fetal blood. So again, the fetal blood is always within the tertiary villi. Maternal blood is always within the intervillar space. Here we can see another example. This would be which one? This would be the maternal blood, right? And this, within the tertiary villi, would be the fetal blood. I think that makes sense. I believe that's it in terms of the placenta. We can finally get to the umbilical cord. One more thing, sorry. We did talk about the chorionic plate, and the chorionic plate is of course the fetal placenta sort of mainland that we have here. And the chorionic plate is made up of the extra embryonic mesenchyme, all of this. Now, if we look at this image right here, this would be the chorionic plate, right? And all of this pink would be the extra embryonic mesenchyme. Now, the extra embryonic mesenchyme is not only what makes up the chorionic plate, it's also what makes up the tertiary villi. So you can see all of this pink area also goes down into the tertiary villi. So if we go down into a tertiary villi right here, all of this white area is also the extra embryonic mesenchyme. Even if we're in the maternal side of the placenta, this being the decidua basalis, not the chorionic plate, because again we're in the maternal placenta, then the tertiary villi here will also still be made up of extra embryonic mesenchyme. You can kind of imagine it as the fetal placenta owns all of these islands as well. So the material that makes up these islands is the exact same material that makes up the chorionic plate. While in the maternal placenta, it doesn't own any of these tertiary villi, therefore it has a very unique material that makes up the decidua basalis, namely the HACPT cells. Okay, getting into the umbilical cord. Umbilical cord also suffers from the microscopy magnification issue, where if we had these images in front of us, it would be a lot easier to distinguish that we're looking at an umbilical cord, because it almost always looks like a little guy either being sad or confused or odd, Picasso formed, right? The umbilical cord is very distinguishable from afar. Zoomed in, it's not. Zoomed in looks something like this, right? Now this is definitely something that appears as any other structure, but to make it a little bit easier, although this looks like we're looking at like a fetal part of the placenta, we will never see all of these islands or tertiary villi in these umbilical cord structures, right? So if we see something that has a clear border and looks like it's fairly a smaller structure with large blood vessels like these, then we're definitely in umbilical cord territory. They all look like this, right? And the umbilical cord has 3 unique blood vessels. It always has 2 arteries and 1 vein, vena umbilicalis and 2 arteria umbilicalis. Now all of these are muscle type arteries and muscle type veins. And the way to distinguish the veins from the arteries should be simple to you, but let's repeat. Arteries have a smaller lumen, so smaller inside, and a thicker wall. So thick wall, small lumen or opening. While the vena umbilicalis has a large diameter lumen and a thinner wall in comparison. So you can see the diameter of this lumen is a lot larger than the diameter of that lumen, right? And the wall of this blood vessel is a lot thicker than the wall of this blood vessel. So that is the main difference between the artery and the vein. So looking into a microscope, we'll see something like
this. Now here you'll be able to hopefully distinguish a vein from an artery. So which of these are arteries? Which of these two is an artery, rather? If you said that this is an artery, you will be correct. Now, why? First of all, we'll look at the wall of the blood vessel. So the wall of this blood vessel is this thick, while the wall of this blood vessel is about this thick. So you see how thicker this wall is compared to this. And another thing, lumen of this blood vessel is this big in diameter approximately, and the lumen of this blood vessel is a lot larger. So that's why we'll know that this is the vena umbilicalis, while this is the arteria umbilicalis. It's just that easy. So if you see a slide where you see like big blood vessels and barely anything else, you're definitely in the umbilical cord area. Another thing, the cells that line the umbilical cord, if we go back here, so all of this border, you can see it kind of darker here. Whoop. Well, that's me trying to explain. This border, maybe this is more clear. All of this is known as something that we have, talked about before, and that is the simple columnar amniotic epithelium. Where did we see the simple columnar amniotic epithelium? We saw it during the during the fetal part of the placenta. That's too far up. Here. So this border is the same as the border that goes around the umbilical cord. Now if we look at this image right here for the placenta, we can see that the border of I love when they're working in the middle of the night. We can see that the border, this is the border of the fetal side, the simple columnar amniotic epithelium, is the exact same border that goes around the umbilical cord. So that's why they're the same. Technically. That's only images. Yes, so we talked about the blood vessels, we talked about the border. Now the thing that makes up the umbilical cord, all of this material, is not known as the mesenchyme, it's known as the Wharton's jelly. It's kind of a goofy name, it's very memorable. If you know that you're in the umbilical cord slide, and they ask you about like the material around the blood vessels, you'll know it as the Wharton's jelly. The third structure that I'd like to talk about is another sort of opening, another sort of tube within the umbilical cord. Now, like I said, we have two arteries and one vein. The vein has a larger lumen, the artery has a smaller lumen, but the artery has a thicker wall, while the vein has a thinner wall, right? But there is a third type of thing within the umbilical cord. That is a gland. The gland has a specific name, and it's known as the remnant of allantois. You may see it, you may not see it. For example, here you do not see it, here you do not see it. So it's not visible in every umbilical cord slide, but definitely something that has been brought up in previous colloquiums. Now, the remnant of allantois is a gland, like I said. And if we remember what I said in the previous video where I talked about the difference between a gland and a blood vessel. So here, if we see this beautiful image that I did, the gland has a border that is thick and looks a lot darker than the border of the blood vessel. The border of the blood vessel is thin, lined by all of these endothelial cells. The border of the gland is thick and dark, lined by all of these simple cuboidal epithelium. Right? So if we look back at the umbilical cord, that's too far. Here we go. You can see that while we have all of these
this thick border. We can definitely see a border here that is a lot more cuboidal in shape, the simple cuboidal epithelium. It will be more visible when we zoom in, so let me find it here. This is an umbilical cord, right? And here they will show you the vein, they will show you the artery, but then they will also show you this. At initial, like the initial line of sight, you will not be able to as accurately distinguish if this is a gland or a blood vessel, which is fair. That is why they will zoom in and further zoom in, so you can definitely see the borders of these cells. You see all of these cells here, how cuboidal they look in shape? That's because they are simple cuboidal epithelium, which means we're looking at a gland, and therefore this whole entire structure has a specific name because we're in the umbilical cord, known as the remnant of allantois. So again, when they zoom in and you see all of these cuboidal looking cells inside, we know that it's a gland or the remnant of allantois. While here, if we look at this vena umbilicalis here, you can definitely see that the cells are very squished and not as cuboidal and don't really have that big shape like the cuboidal cells here. These are endothelial cells, so that's how we know that this is a blood vessel. They will also probably zoom in for the veins and arteries so that you can see the difference. Yes, maybe we should talk about the last structure, which is degenerating or degenerative blood vessel. And it is another opening that just looks sad. Really, it just looks like it's about to fall apart. Like all of this as well. That's just known as the degenerative blood vessel, because we have a clear vein, clear arteries. So this would just be like a sad little degenerating blood vessel. And of course we have the gland here that doesn't look as sad, with the simple cuboidal epithelium. Very easy to distinguish because these have endothelial cells. Here we can see it as well. Again, very sad, barely there, barely existing, barely holding it on. Those are degenerating blood vessels. I think that's it for the class.